Well, welcome everyone. It's a pleasure to be with you, even if it's remotely. This is one of the Boise River Enhancement Network's numerous Riverside chats. You might have known them in the past as our brown bags. We've upgraded and decided that the aesthetics of a brown bag are not as attractive as that of a Riverside chat. So that's what we'll call it from this point forward. It's always a pleasure to share time with you. We, as you know, are natural resource professionals, community scientists, and volunteers working to keep the Boise River a happy, healthy place for all to enjoy. And we're glad to share time with you today. And we're very happy for your interest. I am so pleased to be introducing Tate, and Tate is going to pronounce his last name for us.
Libunao.
Libunao. Contrary to how you might have pronounced it in the past, Tate Libunao, who is a very talented PhD student at the University of Idaho. We have been partnered with Tate and his major professor, Dr. Alan Kolak from the U of I, for the past 3 years in a partnership to study the presence of methyl mercury in the lower Boise River. This is part of a larger study that Tate and Alan have been working on, characterizing the distribution of methyl mercury throughout watersheds in the Pacific Northwest and Intermountain West. And it's an important part of research that will answer the question, is there a public health hazard? And the more interesting question to some of us, why is it where it is and where is it going? So with that, I am pleased to introduce to you Tate.
Thank you for that lovely introduction, Rob. And I am going to make the awkward transition of sharing my screen here. I have a little presentation. And I just want to confirm that everyone can see the screen here. Looks like I get some nods. So yes, as Rob covered in the beginning, myself, I am a PhD student going into my 3rd year at the University of Idaho in Dr. Alan Kolak's lab. For the past 3 years, we've been using crayfish as sentinel organisms to monitor the occurrence and distribution of mercury at a basin level, but also at the more localized watershed scale. So today I'm going to cover the idea of using crayfish as an approachable tool and community-based participatory monitoring. Now, to understand how we got here and why we're doing this in the first place, we must first understand the problem. And the problem is that there is this ubiquitous chemical contamination across the Pacific Northwest. And the EPA in 2009 listed 4 primary contaminants of concern that are of issue in this region, that being DDTs, PBDEs, PCBs, and mercury. Now, there is a challenge with this problem in that the Columbia River Basin, our study site effectively, is over 258,000 square miles, which is a lot of real estate to cover. But the ubiquitous chemical contamination necessitates that scientists need to monitor these concentrations and the occurrence of these 4 contaminants continuously to see where they are and how they move through this environment. So this is at the river basin scale. And if we were to narrow it down to the lower Boise River watershed, we can still see that there's still this challenge of a lot of real estate to cover. It's large-scale environmental monitoring of chemical contamination at even the watershed scale. For lower Boise, for example, encompasses 1,290 square miles with multiple tributaries, the lower Boise River proper, and the connected rivers throughout. It poses this problem of we need to understand and confidently estimate the chemical contamination in these areas, but in order to do so requires a lot of personnel power. It's resource intensive, and it's going to require a lot of sample size for us to confidently say this is what we're seeing in the lower Boise River relative to chemical contamination. So one way that we can meet or close the gap on some of these challenges is by using this tool of community-based participatory science, where essentially what it is where we, in this case being myself and Dr. Kolak, develop the tools that allow the community to then go out and collect the data that we need from the environment in order to make analytical inferences on the current chemical contamination in that location. And what community-based participatory approaches can do from a monitoring perspective is actually increase the spatial coverage that we need to confidently say, this is what that contamination looks like, as well as achieve that magic number of increasing that sample size. So accessing the community in this way can severely help environmental scientists accurately characterize the degree of chemical contamination in the environment. However, there's, I don't want to say an issue, but there's still some challenges that are proposed with using things like community-based approaches. And the tools that have been developed thus far that allow communities to go out and collect this data in the environment is limited to a few usual suspects. And some of the tools that have been developed for community-based approaches allow them to make rapid assessments through either presence-absence data or hot or cold data relative to pH, DO, metals, arsenic, agrochemicals. And this is great. And this has been huge accomplishments relative to these community-based approaches. But there's a whole other subset of chemicals that frankly communities want to and need to be measuring in their environment. Some of these chemicals include things like methyl mercury, like PCBs, like PBDEs, DDTs, and the ever-growing popular compound PFAS. And because of these limitations, it's created this schism, this great chemical divide between what communities can measure versus what they need to be or want to be measuring in the environment. Now, focusing on these chemicals that we need to be measuring, there's a couple different tools or methods that can be employed to gather this information. One of those ways is by using passive integrative samplers, which is just a metal cage with a membrane that integrates a chemical signal over time in the water. And the use of sentinel organisms that the EPA has actually identified a couple different types of sentinel organisms. But again, it still leaves us with this question and this problem of we want to motivate, we want to mobilize and activate community scientists to want to be involved in this kind of work. And frankly, giving them a metal cage and coming back in 2 weeks and saying thanks isn't all that engaging. And on the flip side of using the sentinel organisms listed by the EPA, there's some huge participation barriers that are affiliated with those. Like you don't have communities wanting to go out and trap endangered eagles or coho salmon and taking samples from them, or having to actually sacrifice those animals when it contradicts their moralistic values. So in a way, the tools that we can use to give to communities to measure the chemicals we need, they don't lend themselves to this idea of community-based participatory monitoring. So we have all of these questions and challenges and problems that we need to overcome in order to adequately characterize chemical contamination within the environment. So we had to tackle this problem and start asking ourselves questions. And we needed to find a tool, or in our case, a sentinel organism that can monitor these high priority chemicals, can mobilize and motivate and activate communities to want to come out and participate and gather data in collaboration with us and for us. And it needs to exist everywhere, because our idea is that using sentinel organisms need to occur ubiquitously and wide distribution, because we want to have a bunch of points on a map effectively to where we can make confident estimations on that chemical contamination. We needed to find a sentinel that can be approachable from the community-based participatory approach, where it can also be accessible to all demographics and ages and experience and education backgrounds. So a very low barrier of participation, but more importantly, be charismatic. It needs to have meaning to the people that are participating. And we need to be able to weave importance in a story within our Sentinel that instills value within it. And our answer to this was the use of crayfish as a sentinel organism that can lend itself to this community-based participatory approach, this wheel figure that you see in the top right. So this was what we proposed as our answer to these challenges and problems and gaps. And here's what we've done thus far using crayfish in this capacity. So again, like I said before, our study site is more than just the lower Boise watershed. It's the greater Columbia River Basin, which has a lot of geographic real estate. But in this 3 years time, as Rob was pointing out, we've been able to collect, with the help of people like the Boise River Enhancement Network and other river keepers throughout the basin, over 1,000 crayfish of 3 different species. We've been able to sample over 40 plus sites within 20 plus watersheds throughout the basin. But more importantly, right from the community side, the citizen science side, we've been able to mobilize and collaborate with over 25 distinct community groups, from schools to higher education institutions to nonprofits to tribal natural resource departments. 6 of these 25 are actual tribal entities that we still continuously work with, and we're growing. And over 200 plus individuals from these community hubs have annually participated in this project, which we find as a huge success in developing this basin-wide citizen science network. So we've been able to cover a pretty extensive geography in our project thus far. We've also been able to capture a substantial amount of geographic variation using crayfish as sentinels for mercury in the environment. And what I want people to take away from this figure in particular is that one of our goals is to safeguard public health. And what that means is making sure that people that are going out and directly consuming crayfish have the knowledge of knowing that what they're eating is actually safe relative to mercury, and are in fact following the EPA guidelines relative to consumption of a chemical. Now, what this graph is also showing, aside from that crayfish are virtually safe to eat everywhere, is that crayfish can actually reflect the geographic variation of mercury, indicating their use of looking at their mercury within their body and extrapolating that back to the environment. So they're useful in this regard. We've also been able to ask ourselves, we're using a biological organism, and with using a biological organism, there is associated factors that may influence that animal's mercury concentration. So one of the things we asked is, can we collapse that variation to where we can confidently say what we're seeing in the crayfish is in fact not confounded by things like sexual differences or the mass of the animal. And in our recently accepted publication, we've been able to demonstrate that we've created a standardized sensor, which in our field of environmental monitoring is important when we're using things like fish or things like invertebrates to make inferences on environmental quality. What we've also been able to do is have this mass community mobilization. Thinking back to a couple slides where we've mobilized over 25 distinct groups, we've actually bridged and brought some of the basin together, which isn't easy to do when a lot of distinct community groups have different values, different goals, and different outcomes relative to what they want and what they find to be valuable within our project. So aside from mobilizing the troops to get a great representation geographically of mercury in the Columbia River Basin, we've actually been able to foster relationships among our community groups and almost work as a middleman and networking in some situations. And for example, we work with a lot of tribal schools where we've gone above and beyond just having sampling events with them. And we've been able to have field trips out to our lab where I can demonstrate to the student youth how to properly dissect, how to identify crustacean anatomy, and make it more interactive than just this tier one engagement with the community. And it's been very fulfilling on our end, as well as helped strengthen the bonds of the community of the Columbia River Basin. So coming back to this figure, this community-based participatory research, up until now, we've been able to, if you think of this figure as a wheel, we've been able to turn this wheel a single time, where we've developed the tool of the crayfish to where we can go to the community, give them the proper tools to measure the chemical contamination in the environment, in which we provide feedback to our community hubs in the form of presentations such as this one, or infographics, or subsequent meetings with some of their committee members. And now we're back at this top of, after we've turned the wheel, we want to keep turning this wheel and innovating and redeveloping these tools. And one way that we're redeveloping these tools is we're going back to communities and we're engaging with them and asking them. This is what we've done so far relative to the first turn of the Crayfish Mercury project. What would you like to see done next? Or where do you think that we can go relative to, are there other contaminants in your locale that you're interested in knowing about? Are there other public health issues that you are concerned about within your watershed? And instead of us driving the bus and telling the community this is what we're doing, we're going back to the community and working in collaboration on what other tools can we develop, what other things can we build upon the foundation that we've already done in order to better use our project, but also cater to the needs of the people that we work with and we're working for, frankly. So we ask our question, what's next? And we've gone to the communities and asked, what would you like to see next? And some of these things that we've gone about is thinking about the crayfish not as just a one-trick pony. It's not just used for mercury concentration. And in fact, like I went back to the first slide, the EPA has listed 4 primary contaminants of concern that pose a risk. And using still the same crayfish, we can also look at the other chemicals. We can look at PBDEs. We can look at PCBs and DDTs and its metabolites. And we can start painting a more broad picture with a thick brush beyond just mercury, and including mercury, other metals as well in those systems. And we also can start looking at some of the new kids on the block relative to chemical contamination. One of the ones that you may have heard of, and frankly may have more of a more contemporary knowledge about, is PFAS and its constituents. We can also use crayfish in this capacity to monitor these forever chemicals in the environment, which has gained a lot of traction over the past decade and is only continuing to grow in terms of understanding its occurrence and concentration within our waterways and within our food resources. Beyond just the ones that are listed in the State of the River report, we can also look at a suite of other chemicals that are even more difficult to quantify in the environment. They're difficult to quantify in the environment because they're not going to exist in their parent compound. They'll exist in the biotransformable metabolites. So we can also look at things like pharmaceuticals, personal care products, pesticides, endocrine disrupting compounds that have just as much weight to them and understanding at what concentrations they occur in the environment. We would have to use a different methodology, looking at things like biomarkers of effects, but we can do that. And these are the questions that we come with the community to, is what is of interest to you? Is it personal care products? Is it pesticides being in an agriculturally dominated watershed? These are things that we can use our sensor for to address the questions of our communities. And beyond just looking at the capabilities of what the crayfish can actually do, we're also developing innovative methods to, again, cater to our community hubs, as we recognize that not all communities are made equal, and some are more reluctant to sacrifice a living animal from its native habitat, which is understandable. And in citizen science, quite frankly, it's common that not everyone has the same moralistic values of sacrificing it for science. So we're developing things like non-lethal tissue collections to circumvent or curb some of those worries and lower those barriers of participation. We're also developing things like solution-based approaches that can remain the integrity of our sample if we were to sample from very distinct and far locations, to where we can confidently say that that sample is still representative of that environment, because it's held together the integrity of that sample and the chemical signature within. So as always, right, we're always looking for more participants and more community members to participate in the crowdsourcing of crayfish from across the basin, but as well as the lower Boise watershed. As I said before, the lower Boise watershed is 1,290 square miles, which is a lot of geography to cover. And in order for us to attack that or approach that issue, we need volunteers, and we would love to chat more about that. You can reach myself and my advisor, Dr. Kolak, at our emails listed here. And you can make this collage even bigger, and we'll continue to grow it. And with that, I really appreciate you letting me speak for the second year in a row at the Riverside Chat. It's always a pleasure to see people from the Boise River Enhancement Network, and we look forward to some of our upcoming events on the lower Boise River this year. So I would be happy to take any questions if there are any, or anything like that.
Wonderful. Thank you, Tate. That is informative, and it's a pleasure to be cooperating with you on this project. I need to point out that there are two people on this call who are so important to making our work with you happen. And that would be Ben Everson. Ben, thank you. Shout out and applause. And our coordinator, Marissa Keith, who is so important to having the logistics work perfectly. Any questions for Tate?
I had a couple of questions. Is there, at times I've come across a website for the Mercury project. Can you tell us what that address is?
Yes, and in fact, I will stop sharing my screen real quick, and I will put in our website into the chat box.
Thank you. And then had two other questions. So we've talked before about that you're developing the non-lethal collection methods. Will that be something that you're starting this fall?
That's a great question. And we have ran our experiment to test whether the non-lethal sampling method would be viable. And those tissues are currently being analyzed at our third-party lab. Unfortunately, because of the backloading that the lab has, it may or may not be, the data may or may not be available for us to analyze in order for us to say, oh, this will work or this won't work. So to avoid any kind of assumptions, as of right now, I'm going to say the non-lethal sampling method approach is not something that we plan on doing this fall. However, We just want to be transparent with that. But however, once those data are in, we will actively and quickly analyze to see if that is a viable method. And if that happens to be before our events that are going to be carried out this fall, I would be open to at least trial it. But if we are looking to still, I'll say confidently estimate the mercury in certain tributaries or unique locations that we have not sampled at yet, I would still caution against that and recommend that we do it the traditional way that we've done it in the past, which is taking the whole crayfish, humanely euthanizing them, extracting all of the tissues that we can, as they're all valuable, and looking at them from that way.
And then the last question I had was, there is a large crayfishing community here in the Treasure Valley. They have a whole Facebook page dedicated to showing off their catches and giving advice and everything. If those people wanted to participate maybe outside of one of the official events, is that possible? Like, could they get the crayfish up to you some way?
Oh yeah. So, and if we're on the same page here, Marissa, I believe we're talking about the Idaho Crayster, or the Craysters of Idaho. It's a Facebook page that is predominantly within southern Idaho and the Butte of Idaho, Pocatello down to Boise or Nampa, and huge following, which I am a part of, and I actively try to post on there to either to see where people are catching and what they're catching, and kind of gauging the field that way. If they wanted to participate either within the event or independently, I would say get in touch with myself, or I would advertise to those individuals to get in touch with myself or Dr. Kolak, to kind of give them the once over on if they were going to participate independently of a sanctioned event, that they make sure all the T's are crossed and the I's are dotted in terms of how many samples we need, how to store, what information we need relative to those crayfish, because I wouldn't want people to have to waste crayfish and them not be usable within our data set. But they're more than welcome to participate independently. And in fact, and I don't want to get ahead of myself here, and I've talked about this in the past, of developing a method or a kit, a toolbox, in which allows communities or individuals to independently sample for crayfish, which right now is still in queue when we're waiting on this non-lethal sampling method, because that would be the answer to a lot of those questions that you asked previously.
Right, thank you.
Mary, I'm wondering if you have any questions.
Hi, this is Ben. I've got a couple of questions.
Hi, Ben.
Perhaps we could reach out to the Craysters or some of those folks to just have a representative at our sanction events. They'd be great teachers, could suggest sites. And then I'm going out next week to look at a site lower than we've done before. A friend talked to me about, and I also want one above where we've done. And so once I do that next week, we should probably try and nail down a date, or at least that we're moving to. I'm going to be gone part of September. So, yeah.
Yeah, that's actually a great recommendation, Ben. I've contacted the person, I'll call him the founder of the Craysters of Idaho, who also makes the traps and the attractant and everything.
Right.
And apparently he's partnered with a bunch of people either at the university or Boise State who have been interested in crayfish. And I've had some back and forth communication with him, but I think it would be a tremendous idea to get somebody from that administrative side of that page and come down and either suggest sites like you're saying, or even act as a volunteer stewarder of crayfish facts. Because Lord knows I can't hold all of the facts about crayfish in my head. And it may be valuable to have someone who does it as a recreational hobby and sport.
Yeah.
Yeah.
Okay.
And relative to different sites, I know myself and Rob have talked about a couple adjacent or reference sites that feed into the lower Boise that may have some crayfish within them. And I think that finding additional sites beyond what we've sampled already is highly valuable. And even beyond just the events, again, independent sampling can be carried out just with the proper a priori, I'll call it training, or at least knowledge of how to catch and keep the crayfish, as well as the information needed.
Okay. Thank you.
Thank you. Rob, you may be muted.
Yes, I am. Alan, I'm imagining that you are the keeper of the questions that we didn't ask that we should be asking. What's top of mind for you?
That's a great, Rob. That's a great question, and I think getting back to something that some of us have talked about previously, which is one thing that we would like to hear from the community, whether it's Bren or the Boise River watershed community writ large, is this is a problem that we have. Can we use this model to address this problem? And if so, how? So that's where Tate and I would really like to hear from you all. Not necessarily today, but we'd like to hear from you all what's coming down the pipeline that is a monitoring concern. And yeah, I'm specifically thinking about PFAS when I'm saying this, but a monitoring concern that might potentially benefit from what we're doing with crayfish. Now, PFAS, and I can say this because, frankly, I'm old, right? And what I mean by that is PFAS is the chemical du jour. It's a chemical of the day. PFAS is going to be here forever, just like DDT is here forever. But it's not going to be there are going to be new chemicals. There are going to be emerging chemicals that come on the scene every year, every decade from now on. And I think it's really interesting, as Tate was alluding to in the talk, I think it's really interesting that, can this system be used for pharmaceuticals and personal care products? Yeah. Can it be used for endocrine disrupting compounds? Yeah. Could it be used for, I don't remember the other compounds on that list, but sure, it could be used for a variety of different chemicals. The important thing is every chemical has a suite of physiochemical characteristics that by having such characteristics, it determines the best way to approach it, either using an animal like a crayfish or just measuring it in the water. And so we, the Idaho water quality community writ large, we want to have a toolbox where the appropriate tool can be used at the appropriate time. So I think that's where this is ultimately going, and you're capturing a young man in Tate, where as you were saying earlier on when we started, Rob, he can be around for a long time, way past my time here, and be stewarding that type of an interaction. So I think from the state of Idaho perspective, it's really pretty exciting.
So part of what we're doing here is recruiting a new generation of people like you and I.
Yeah, that's exactly right. And that's not just Tate, but and you think about it, right? And all of the K-12, college age, whatever, the youth that are involved in this project and other projects in the Boise River and in other rivers across the state. Yeah, that's exactly what we're doing. is we're doing that. And in today, 2024, or I guess it's still 2023, if those people happen to be tribal members, if they happen to be African Americans, if they happen to be Hispanic, if they happen to be, first generation college, so much the better, right? I mean, so much the better from as many individuals say, right? The diverse workforce generates the better answer, right? And we can help with that. And by doing these type of outreach activities with Idaho youth and Washington and Oregon and Montana youth as well, but specifically doing that with Idaho youth, hopefully we can generate interest in STEM fields that might not otherwise be generated.
Thank you, Alan. I have a question for both Tate and Alan, and that is, what is it that we, and in this case, we being the Boise River Enhancement Network, do to be a better partner, to cooperate with you to make your project a success?
Well, I'll start by saying that our continued cooperation has already attributed to our success, both in the form of publication, data collection, as well as relationship building. Relative to this project, I think one thing, and kind of echoing Dr. Kolak's answer here, is if we can diversify and increase the amount of people, participants in these types of collections, and this is beyond research, right? We can actually start making local impact on providing hands-on opportunity to student youth, to underrepresented communities. And I think that is one way in which we can actually make substantial impact, change, and increase our success beyond of what we've already done thus far. That's from the project's perspective. From a research perspective, again, kind of echoing Dr. Kolak, there's seemingly right now no limit to what kind of chemicals, where using a sentinel organism is appropriate. There is really no cap, it seems like, on what we're able to monitor in the environment. So gauging what kind of chemicals the residents of the lower Boise watershed are interested in, or what they want to see, would be very valuable to us and actually help us make the case of bringing this project beyond just Mercury and Crayfish. And I think that's one way in which we can increase those parts.
Yeah, and I think Tate did a really nice synopsis of effectively our approach or our vision down the road. And I'd just like to embellish upon that a little bit from the perspective of what we would really value from relative to everyone on this call today, but also everybody within Bren, the individuals, the other stakeholders that you all interact with. What we would really value would be input from, and again, looking back at like the community-based participatory research approach, what we would really value would be hearing your harebrained ideas, frankly. So if you said, like, I've been thinking about it, and what would be really kind of interesting is if, and I'm just, totally making this up, but it'd be really kind of interesting if we took a reach of the river and said, okay, let's look at the salmonids, the crayfish, the bivalves, and the paraphyton, and how would we go about doing that? And what kind of questions could we ask, and how could we go about doing that? So I think, kind of getting back to what Tate was saying, looking at it from a research perspective, it would be really valuable for us to hear from the community relative to what I just eloquated, where the ideas are really yours, and you're coming to us with a question, and then we can figure out we can sit down with you and work out an experimental design or whatever else. But I also think today, and again, given that it's 2023, and again, I'm an old guy, I've been at this for a long time, and 10 years ago, I can promise you we would not be having this conversation. But today, I can also promise you, as long as there's, and I'm not trying to get political, I'm really not, but as long as there's a Democrat in the White House, we're going to be having these conversations. Even in a red state like Idaho, we're still going to be having these conversations. And that's, if there is interest from, let's say, Bren was to say, hey, we've noticed our demographic, and it's all older white people, right? That's who come to our things. How do we make this more welcoming to other members of our community? And I'm not trying to put words in your mouth, and I'm not saying you should do that or shouldn't do that. I'm just using that as an example, right? I think we'd be really interested in those kind of questions. And frankly, speaking of, I can speak to that wearing two different hats. One hat is community scientist, and my career, if you look at the course of my career, the last 3 or 4 years, I've done a whole lot more work with communities than I ever did in the first 25 years, which is to say my first 25 years I did none. But now I'm doing a lot of it. Part of it is because, again, I mean, I know, and I'm not trying to make this about me, but part of it is because I'm getting older. But part of it is because funding is available to do that kind of, to do those kind of outreach programs. So I think we're in a position where if those kind of things are of interest, like I'll give you an example. Tate and I have been talking a lot, right, Tate, over the last 6 months about, there are forums in the state of Idaho that bring K-12 kids together and under the auspices of a science fair. I've been to a few of them, not many, and I'm not making any judgment about the science fairs that I've been to, but it would be really interesting to have a science fair with significant tribal representation. Partially because it's valuable to get the tribes involved, but secondly, imagine both the cultural as well as the scientific cross collaboration that a bunch of tribal kids could have with a bunch of non-tribal members. I think that's just a really, really cool thing. And that may not be that important in a state like Maryland or, New York, maybe. But in a state like Idaho, where we've got, how many different tribal nations in the state, it's really, really important. And I think that's something that if you guys were coming to us saying, hey, that is something that we would be potentially interested in, that's the kind of thing that Tate and I could help with.
Wonderful. You have touched upon another important question we've been asking ourselves, and that is, how do we get Indigenous people involved in our work? And excuse me, we'll be working in the next year to involve the Shombanek tribes in particular.
Now, I'd just like to, I'm going to, I guess in a way, and I mean this in a very positive, gentle way, I'm going to throw Tate under the bus relative to this one, because we just had a conversation, right, Tate? We were talking the hour before Tate gave his presentation. We were talking about Tate and professionality. And Rob, and I'm not trying to, trying to force Bren's hand or anything like that. But if you're going to do that and you're going to start having kind of conversations or meetings with tribal members or how are you going to do it? Both myself and more importantly, Tate would absolutely love to have a seat at that table, because imagine what that does for Tate's professional development. And I'm going to talk about him even though he's right, on our Zoom with us all. But I mean, that's that, as you know, Rob, that is invaluable for someone's professional development. So I think those are the kind of things that and again, just using that as one example, but that's the kind of thing that we'd like to hear from you, Bren, about. Hey, look, we're thinking down the lines potentially of PFAS, or we're thinking down the lines of diversification. Is that something that the University of Idaho can collaborate with us on? And the answer is certainly yes. So, yeah.
If I were to be thrown under a bus, that's the kind of bus I'd want to be thrown under. Thank
Exactly.
you. I can answer, and I'll dare to mix my professional work with my pleasure, which is working for Bren, by saying that, yes, I have clients who are very much interested in knowing more about the processed water that they discharge to Indian Creek and the potential of introducing forever chemicals in that processed water and ways in which it could be perhaps removed. So I see research opportunities for us to work on together that include that. I want to finish by asking one final question for Tate. I mentioned a suite of water quality data that's available from the U.S. Geologic Survey that's contracted by the Boise River Lower Boise River Watershed Council. Some of that data includes an assessment of benthic organisms, mostly diatoms and different algal species growing on rocks. How would you use that kind of data with the data that you've generated to answer questions we haven't even thought about?
Well, it's kind of a complicated mix, right? Because there's a few things we can ask, and it kind of depends on the nature of the data, whether those data are associated with things like mercury, for example, or if they're associated with, like we've had conversations in the past about fluxes of nitrates or phosphates that can contribute to heavy algal blooms and have downstream cascading effects like lower DO. If we see a similar pattern trend with mercury increase in crayfish following or correlated with those inflection points. And that's one thing off the top of my head. But again, right, I would need to, and Dr. K mostly as well, would have to sit down, look at the data that's available out there and start asking ourselves the question, what can we use the available metadata for relative to crayfish and mercury? For example, we're actually in the process of working with the Spokane Tribe Natural Resource Department, as well as Washington Department of Ecology on the Spokane River, where the Spokane Tribe is interested in looking at the relationship between sediments, sediment type, biofilms, and other benthic organisms in mercury, as well as crayfish, because what they're finding is that not all of their sites have available biofilms or non-detects of mercury in the sediment. And they're seeing if they can use crayfish, because those are present where the other information is not, if they can use crayfish as a proxy to back calculate some of those environmental concentrations, as well as looking up through the food chain. What's the relationship between crayfish mercury and fish mercury? So the Department of Ecology is running a fish study to characterize fish consumption risk along the Spokane River. And we're actually interested in asking, is that relationship consistent across fish species or location? And can you in fact use crayfish as proxies for fish mercury to act as a pseudo consumption risk, where catching fish may be more logistically challenging? I could see that in the Boise River, we may be able to apply some of those similar questions to there. But again, given the nature of the data that's already available. which has led me to ask people with DEQ as well as the city of Boise for historic metadata collected on the lower Boise River relative to mercury or phosphate data.
Very good. Any final questions for Tate, Alan, or anyone who might be on this call? Okay, well, this is one of a series of Riverside chats that we have planned for the upcoming 6 and 12-month period. Marissa, would you tell us what some of those future events might be?
Yeah, so we have a couple of volunteer opportunities coming up. The first would be on Friday, August 4th at 9 a.m. We're doing a Your Mother Doesn't Live Here Boise River cleanup. It's the first river cleanup with that name, thanks to Rob. And so we will be meeting 9 a.m. at Ann Morrison, entering off of the west entrance and then parking by the river. But there will be info on our Facebook page or the website, which is boiseriverinhancement.org. And then the Crayfish Mercury study, I believe we have a tentative date of September 9th, but we'll check and make sure and get all the logistics for that figured out. And that will also be posted on the Facebook and the website. And so that is a community event. Families, children are welcome. And we usually do that in the morning hours before it gets too hot. And then we do also have a rafting trip opportunity coming up. We've partnered with the Boise River Outdoor Opportunities. They are doing a benefit raft trip with Bryn. And so that info will be up for the public to sign up if they wish to do that this afternoon.
Thank you
And that
very
will
much.
be August 8th.
And as always, if you like what we're doing in the public interest and find value in our work, and if you have some change perhaps jingling in your pockets or sitting on the floor mats of your automobile, we would be happy to accept any and all donations to our work that you might be willing to share with us. Thank you, everyone, for being with us. We look forward to seeing you at our next event. And thank you to Tate, to Alan Kolak, to Ben Everson, and Marissa Keith. Have a great day.
Thank you, Rob.
Bye, everyone.